Subscribe to our YouTube channel and press the bell icon to get the latest updates.
I'm sure many of us heard about the spinach plant, which has been modified to be able to send emails to humans. This was a 2016 experiment from MIT, but it has been making the rounds over the past few days. In the past, we've also quote unquote taught plants how to take selfies, and there are many other things that we have been experimenting with plants, whereby we're using plants' natural functions to our advantage and to enable them to kind of communicate with us. All of this falls under the field of plant nanobionics, about which we will be talking in this video. I'm Sandhya Ramesh and this is Pure Science. Plant nanobionics is a new field of bioengineering that inserts nanoparticles and devices or sensors into the cells and bodies of living plants. This can either modify or amplify the natural functioning of a specific region of plants or a specific organelle or tissue. The bigger objective is to create a monitoring system whereby plants can themselves monitor their environment, power their own devices with their own energy from photosynthesis, and perform communication with humans by triggering other devices which can in turn send signals, or in the case of the spinach, emails to us. So let's look at this spinach example itself to understand how it worked. This was a 2016 experiment at MIT where ultimately researchers were able to use the spinach plant to obtain an email alert when toxins built up in the soil where the plant was. Now, how did this work? Toxic compounds called nitroaromatics, which are found in industrial waste and explosives, often seep into the soil through water and groundwater. This then enters plant roots because these compounds and molecules are about the same molecular size of nutrients found in the soil, which the plants also absorb into their bodies through their roots. If you remember, in school we studied about xylem and phloem, how the root takes up water from the soil and carries it through the plant. And that's what happens here. The root system carries the water from the soil, and this gets transported up through the plant and reaches the plant leaves through the xylem. Now, plant leaves are where photosynthesis occurs, but it's also where transpiration occurs. Transpiration is loss of water in the form of water vapor from leaves through small pores that are naturally found on the leaves. Of all the water that a plant absorbs, it ends up using only 5% for its own growth and for its own processes. The rest of it escapes as water vapor through plant leaves. Now, as the process of transpiration or water loss occurs, the chemicals that were transported along with this water build up in the leaves and get accumulated there. This accumulation occurs specifically in mesophils. Mesophils are the tissues that are present in the central thick body of leaves. In a leaf structure, you have the epidermis, the top layer and the bottom layer, which have the guard cells, and the stomata, which are the natural openings through which oxygen comes out. Everything else that makes up the bulk of the leaf is called the mesophyll. It is the body of the leaf. So as this chemical buildup occurs in the mesophyll, it can be monitored. To do this, MIT engineers implanted single-walled carbon nanotubes inside the leaf mesophyll of this spinach plant. These were also equipped with fluorescent nanosensors. Now, as nitrogen dioxide, the toxic pollutant which was being monitored and tested in this experiment, built up inside the leaf, a signal is emitted above a certain threshold of concentration in the mesophyll. This signal then triggered a pre-programmed email alert. This alert helped scientists know that the leaf concentrations of the nitroaromatic, and thus by extension the amounts of it in the soil, crossed a certain threshold. This is one use case by which the soil content and pollution can be monitored directly by plants. Because plants are so quick to react to environmental changes, many researchers believe that such plant nanobionics could be used to monitor the environment, with living plants themselves acting as active monitors around the globe. While this experiment was performed to detect explosives, plants can also be used to monitor changes in the climate or in pollution levels in the surrounding area. Plants are very apt and very uniquely suited to perform this kind of large-scale global monitoring because, first of all, they grow in places that humans don't live in or frequent. So they can perform observations in places that we regularly cannot. Additionally, they can generate their own energy from sunlight through the process of photosynthesis. So they can in fact power themselves constantly without us having to generate or replenish energy for powering these kind of sensors or other devices that are used in the field of plant nanobionics. It is in fact using energy from photosynthesis that a plant called peat was taught to take selfies. For example, a maidenhair fern, one of my personal favorites and notoriously difficult to grow, was able to snap regular pictures of itself and its surroundings using energy generated by photosynthesis. Now we know that photosynthesis produces oxygen and glucose. Oxygen is released into the air through stomata, but glucose travels around the plant and also travels back to the soil through roots as a waste product. In the soil, there are microorganisms and bacteria that then absorb this glucose and further break it down, releasing even more energy in the process. This energy can be harnessed using microbial cathodes, anodes, and a capacitor, if you remember how batteries work and store energy from our school physics. The microbial capacitor stores energy, and when it is full, it discharged power of about 0.1 milliwatts, and this triggered a camera. This experiment was performed at the Zoological Society of London in 2019, and the plant and microorganisms were able to snap pictures every 20 seconds. There are text stories for these, which will be linked below, of course. But imagine a plant being able to take a photo of its surroundings every 20 seconds. That is a big deal and offers us incredible scope for visual monitoring of forests, for camera traps, and of course monitoring the environment in ecologically sensitive areas where we can't keep going again and again regularly. So this could aid tremendously in conservation efforts, as well as to track and monitor other kinds of animals that are hard to track otherwise. Furthermore, what's special about these microbial cells is that while the plant itself needs sunlight for photosynthesis, these microbial cells are present in the soil and they work in the shade. So they don't really rely on sunlight or any other form of external energy to function. These were two of the more popular examples in recent times of how plant nanobionics work. Nanobionics is the study of structure and functions of a biological system like a plant and the use of engineered materials and machines at a nanoscale level to work in conjunction with these biological systems by using their own natural processes. These systems don't need to function just for our benefit. They can also be used to benefit the plants themselves. For example, in a 2014 study, researchers were able to use, once again, single-walled carbon nanotubes to enhance photosynthetic activity. This enabled the plant to increase its own energy output. They also showed that these carbon nanotubes, which enable fluorescent monitoring of nitric oxide, as we saw in the spinach experiment, can be used to function as a chemical sensor. Researchers, once again at MIT, have used a solution of modified, negatively charged, DNA-wrapped nanotube particles, which were further wrapped in polyacrylic acid and rubbed onto the underside of the leaves, where the stomata or the natural openings of the leaves are present. This liquid then seeps into the leaves and passes on into the chloroplasts, where chlorophyll is stored. Chlorophyll, as we know, is responsible for photosynthesis. This helped the plant capture more energy from sunlight. Plants typically tend to use about 10% of the energy that's hitting the leaves. But in this experiment, the leaves were able to harness wavelengths that they normally couldn't, increasing the electron flow in the plant by 49% and thus increasing the energy output. We could potentially use plants like these for energy harvesting in the form of light harvesting, and also use their own natural functionalities to monitor the environment in terms of things like pesticide use, or discover new fungal infections or toxicity that is being induced by bacteria in the soil, and so much more. They can of course monitor the plant's own health as well, helping us conserve more plants and protect more crops as food security becomes an increasingly problematic issue in the near future. One of the leading places where plant nanobionics is being researched and experimented with is MIT, where a lot of these studies are coming out from. But there are also other places and private players that are now picking up on this engineering. And this is a field where we can expect lots of development and many news articles in the near future that attribute anthropomorphic or human-like intelligence to plants over the next few years.